Welcome to bio class. Today we will do one experiment, one practical, which we will perform in laboratory. But before that, we have to know various part of it. Here is the first experiment which is here. That is the study distribution of stomata in leaf. Stomata, I think everybody you know. Distribution of the stomata, it indicates that it is not equally distributed. That is why there are two different type of distribution. Suppose if I draw here one leaf. So here if it is upper surface, then another the lower one is the lower surface. So upper surface and the lower surface, in both the surface, stomata is available. But we know that the lower surface, especially in the dicot leaf, the lower surface stomata is much more than upper surface, because upper surface is blocked with a palisade parenchyma, so that the space is less to open the stomata. In case of monocot leaf, that stomatal distribution upper and lower, it almost equal. So all these things you when will do that, will able to understand. Here in practical laboratory, how we can do it, that at first we have to learn it. And about the theory part and all these things, here I am not giving stress, and I think you know that what is stomata and how stomata it looks like, the stomatal pore, the guard cell, subsidiary cells, and how it opens and closes. So come here, how we can do in practical. So before doing in the practical in laboratory, you must see this video carefully, and so that you can do nicely there. Here our aim is study of the distribution of the stomata. Distribution of stomata means the in upper surface the number and the lower surface number. So number, the number is numerous. So number will not count. Just we can say that it is more and it is less. All right. Now, what kind of materials is it is required? If you can see it in a little bit large view. So here it is. We need one leaf. See that here I am not telling about any name of the leaf, because that name we all the time cannot provide you. Thick leaf. The leaf should be thick, so that we can very easily peel off the layer. Okay. So after that, the brass is required, the microscope, of course it is required, petri dish, slides, glycerin, watch glass, safranin. This safranin, it is the staining solution. Will stain in that. Okay. So let's see that how it can be done in practically. Okay, look at here we have taken one leaf. Leaf, you can see the two clear sides, that upper, here it is lower side. If we want to take it out the lower epidermal part, so you can see this areas, it is here I am peeling it. So if you want to take it out that upper side, then you have to take the leaf, the upper side in front of you, so that the upper part will get that. So upper side and the lower side, that practice you little bit you to do. Now safranin is the solution. Safranin, that staining material, if we stain it, the upper and the lower, then we can see it better, nothing else. And here we are transferring the supposed lower to the water to wash it. Extra stain should be washed properly, and after that it is kept in the slide, the middle of the slide, Look at here, it is kept in the middle of the slide. Then here the glycerin is the temporary mounting media. Temporary mounting media, the glycerin we are, and the cover slip we are over that, and no air bubble should be there. And the extra glycerin which is found outside, it is we are wiping it properly. Exactly you have to do in the laboratory. So that is the one of the upper lower one, and here the upper one we are taking and doing the same, the washing in the water, and we are transferring it in the slide, and here the glycerin mount, glycerin media we have to give, mounting media, and the cover slip we have to clip it in such a way that no air bubble and no gap should be there. So it should be clean, neat, and we'll wipe it in such a way. Now we have to ready for the observation. Here is the microscope. We can clip it inside the and here is the upper epidermal. The stomata. It has lesion number, and another slides also we have to see simultaneously, side by side. In it is in the lower one is, if we compare it, the lower one is more stomata than the upper epidermal. Here is the stomatal view, you can see. Okay, so in this video, if we see it once again, that part, the first part once again, how we can do it. This part is a little bit complicated, others part are not. in part, first we have to check that the upper and the lower surface. If we want to peel it the lower surface, then lower surface should be front. See that, then my front is lower surface. Now I am tearing it, tearing in such a way that the peel will comes out. This is the peel. So see that, and very small part is required. So don't worry if the very small part will come out. And if I do the upper, then upper fish will be there, and and this area then you will transfer it. So this is the practical. You know that from the video only we can guess that how we can do that, but actually you have to do it in practical. So before going to the lab, very soon we will go to the lab. Before going to the lab, please watch this video carefully, so smoothly you can do it there. Thank you.